Hello. Hello.
Hi, how are you?
Uh, so I am Lilith Bharadwaj Baru, sir. So I'm from India. I want to give a little introduction about my one of my friend Mehul Arora. He just gave me the link to join this meeting. So I want to give a little introduction about why am I interested to join in this project, and so I want to give a little details about that.
All right, that sounds good.
Yeah.
Go ahead.
So basically, I completed my bachelor's in VNR, so one of the Indian institute. So where I did some part in the medical imaging. So during that time, computer vision, medical imaging was very much intriguing to me. So what I did is I've tried to understand how these neural networks develop for the systems, like the neural network, suppose. So how they have approached, and how is this biological vision actually fundamentally processing. And I wanted to understand, is there a relationship between the biological vision and the computer vision, what approaches that previously it has been done. So I was no so currently I am in doing my masters in IIIT Hyderabad under Professor Bapi. So where I'm trying to understand human cognition specific to how the visual data, for the given visual data, how the brain actually works. It starts from like retina there, and then LGN, and it goes back to visual cortex. So I'm trying to understand so how the mechanisms, how these interconnected neurons have been developed, and how the current neural networks are like working. So I'm trying to bridge between these two. So my interest lies in that, and I have studied a little bit in principles of neural design, the way they have created certain good principles. The book by uh, Peter Sterling and Simon Long. So my little interest lies in these areas. So,
That sounds
and
good. Yeah.
yeah, and I have seen from that book I got inspired. They start from like simple small this nematode C. elegans, where it has smaller structure of neuron, but it how it environmentally, right, it actually works similar to that of having like 302 neurons. How is it able to actively participate in the environment, and how the behavioral changes happens in human cognition? It becomes a larger scale. So I am trying to understand, so how the neuronal connections are processed in human brain and different organisms. And yeah, so bridging computer vision, how the computer line of speech is, and how biological vision is. So this is like current my interest.
Yeah, that sounds great. Yeah, so we've done a number of things in those areas. We're interested, of course, in C. elegans connectomes and C. elegans development, both in terms of the connectome and morphogenesis, and then linking that to computational models. And yeah, we've done some work on comparing biological networks and artificial networks, neural networks. So yeah, we should get in touch a little bit more. So yeah, it's good. And then our group, we have people like Susan who are interested in developmental biology, and Morgan who does a lot of neural modeling, and he has a background in neuroimaging. And of course, Mehul does a lot of stuff with networks. We have a lot of people who can you can talk to as well in those different areas. Okay, yeah, that's good. Thank you for the introduction again. Right. Yeah, so welcome Soham, welcome Lilith, Susan, Morgan. Any other things before we get started, or Any other updates? Okay. We had some things in the chat. Oh, just some. Okay. Yeah. So, like I said, this is the Google Summer of Code period is coming up. Oh, there we go. Thank you. I sent some messages, or I sent the people at INCF a description of the projects. Actually, there's only one project for DevoWorm, but like for our general group that I'm leading or I would be mentoring. And I don't know, we're pretty early in that process. But, we're going to try to follow up on the projects from last year. That means that we'll have some space for the hyper networks, hypergraphs, some space for the growing GNNs, and maybe some combination of the two. So we'll have to see, what people come up with on that. And everything comes out of the repository that we've been working from. That's the DevoGraph repository. That's at github.com, and then of course we have the DevoLearner organization, and then the DevoWorm repository. So everything can be found there. Okay, let's get started. So today I want to talk about two things I've found, two papers. The first one is this paper, and this is, I guess it's some sort of theoretical comment that I don't know what journal it looks like. It's supposed to be in a journal at some point, but this was put on the arXiv around the end of last year. This is Jesus Marco de Lucas from Spain. They're from an Institute of Physics in Spain. And its title is From Worms to Mice, Homeostasis May Be All You Need, which is, if you're familiar with the machine learning literature, there was an initial paper called Attention is All You Need, and then everyone kind of did a play on that title. So that's what they're referencing there. And then this is, worms to mice. This is a cross-species comparison of what they're calling homeostasis. And the point is that homeostasis is an explanatory factor. So let's read the abstract and see what they're kind of getting at here. So in this brief and speculative commentary, we explore ideas inspired by neural networks and machine learning, proposing that a simple neural XOR motif, exclusive OR motif, it's the exclusive OR logic gate, involving both excitatory and inhibitory connections, may provide the basis for a relevant mode of plasticity in neural circuits in living organisms, with homeostasis as the sole guiding principle. So they're going to do, I guess, is look at different neural networks in different organisms, and then look for this XOR motif, and then this includes both excitatory and inhibitory connections, and that's sort of the basis for all of these different connectomes. This XOR motif simply signals the discrepancy between incoming signals and reference signals. So this is where you have this logic gate at this in this circuit, where you have this ability to sort of figure out the difference between incoming and reference signals, and that's why you have this motif. So if it serves some functional purpose, and again, this is just the way that they're describing it. It's not that the brain is implementing some sort of computational motif, like as it's just kind of the way we can describe it, not necessarily that there's an inherent thing about that computational rule. Thereby providing a basis for a loss function in learning neural circuits, and at the same time regulating homeostasis by halting the propagation of these incoming signals. So they go on to say the core motif uses a 4-to-1 ratio of excitatory to inhibitory neurons, and supports broader neural patterns such as the well-known winner-takes-all mechanism, or WTA mechanism. We examined the prevalence of the XOR motif in the published connectomes of various organisms with increasing complexity, and found that it ranges from tens in C. elegans to millions in several Drosophila neuropils, and more than tens of millions in mouse V1 visual cortex. So this is where you have like these motifs. You can look at the connectomes. Of course, C. elegans is the smallest. It's about 300 neurons. Drosophila neuropils, which include thousands of neurons, and then tens of millions of neurons in mouse V1 visual cortex. So you have a lot more than cells than that, so you have tens of millions of these motifs in visual cortex. We'll see if they propose it at scales. I don't think you can really say much from maybe a couple of data sets, but suffice it to say, the more neurons you have, the greater number of motifs you have. If validated, our hypothesis identifies two of the three key components, an analogy to machine learning models, the architecture and the loss function. So this is where we look to these connectomes, we look for these computational motifs. They allow for information processing locally in the network. They then have these attributes of, say, like sort of a loss function, and then also some other type of comparator role in neural computation. And then that's like analogous to what we see in machine learning models in the way that handles learning. So we can assume that that's a learning mechanism in those connectomes. So we have the architecture and loss function, and we propose that a relevant type of biological neuroplasticity is simply driven by a basic controller regulatory system, which is persistent and adaptive despite the increased complexity of organisms throughout evolution. So again, as we get these different size connectomes, we have this neural rule that's sort of, constant across these connectomes. So I think this is a nice paper because it's short and it lays out this hypothesis, which is always, brevity is always better, but it also gives like these comparisons across different connectomes. So the first thing to do is define homeostasis. So homeostasis is a general term that can be defined as the ability or tendency of a living organism, cell, or group. So in this case, we're interested in a group of cells, a network of cells. So this network has a tendency to maintain the conditions within it and at the same level, despite changes in the conditions around it. So this means that when your connectome is challenged by its environment, and especially in this case, by some learning task, if the organism needs to learn something, then the connectome has to change and has to encode that information. But the problem is it can't change too much because then it falls out of equilibrium, I guess, the functional aspects of it. So, you think about that at the level of synapses, weights of the synapses have to change. And when they, but you don't want to disrupt, all the things that the network has learned before or its general function. In our Saturday meeting, which is our Saturday morning NeuroSim meeting, Morgan and I were talking about this paper where they used spiking neural networks and introduced the concept of not just weight matrices for a neural network, but like delay matrices or temporal lag matrices. And I think given this sort of mention of homeostasis, that's worth going back to think about. That, neural networks aren't just about, connection weights and looking at how those change through learning. But there's also a lot of temporal delay that occurs. And especially in circuits where you're trying to selectively inhibit things and selectively, um, reinforce things, that this can be like, an important mechanism. So you want to be robust to fluctuations in the environment, undue change, but also the state of internal balance, which goes back, of course, to Cannon and talking about homeostasis as this general mechanism for physiology. So there's this internal balance again in the network that has to be maintained. So that's homeostasis. However, in the context of neuroscience, in addition to conditions at key levels, such as intracellular calcium concentration, the term also refers to the equilibrium achieved in the propagation of electrical and chemical activity, preventing unstable conditions such as neural hyperactivity. So basically something like having a seizure, where there's uncontrolled excitatory signal sent across a certain part of the brain, and you get seizures instead of normal function. So you want to be able to, apparently what happens in the brain is you get this inhibition, you get this excitation, and there has to be a balance of the two in order to get proper function. So that means that these networks have to have motifs that enable the sort of regulation of this balance. And so that's what they're kind of talking about also with respect to homeostasis. So they had another short commentary. This is the link here, reference 2. We'll get to that in a minute. We presented a basic neural motif composed of a few excitatory neurons and one inhibitory neuron, corresponding to C. elegans, which is basically, you'd find this neural motif in C. elegans, and showed that it can implement an XOR switch, exclusive OR switch, such that when it's comparing two input signals, its output is non-zero, only if these signals are different. So this is exclusive OR. So you have two input signals, you compare the two input signals, you have to give an output. The output is either 0 or it's non-zero. If it's 0, then this means that the two input signals are the same. If it's non-zero, that means it's different. So if you have like a 0 and a 1 passing into this logic gate, then you get an input of non-zero or 1 when the signals are different. If the signals are the same, then you get a 0. Okay, so it's basically, you might get like, if you can replace the 0 and the 1 with inhibition and excitation, then that's basically the motif. We also described how such an XOR motif can be used in a computational neural network as the core of a basic autoencoder and trained using the so-called liquid time constraint or liquid time constants neural network framework, or LTC framework. In this new note, we extend the previous implementation to the case of spiking neurons. So again, we're dealing with spiking neurons, not just like a standard neural network. And explore in more detail the presence of this XOR motif in different organisms using the available open data for their connectomes. So there are a number of open data sets for the new people we know, or the people who've been around here enough know that there are all these open data sets for connectomes. You can download them and reconstruct a connectome, but that only gives you the connections. It doesn't give you any more information than that. You can then play around with the connection data, some neurophysiological data, and actually come up with some interesting things about the connectome. So first we examine the C. elegans connectome again, but requiring the explicit XOR motif to inhibit connections. Then we look at the recently published connectomes of Drosophila. There are several connectomes of Drosophila for different parts of the Drosophila brain, and those are available. They're a much larger size than the C. elegans connectome, and people usually analyze those piecemeal. They usually look at, say, like there's usually some sensory connectome, for example, and they'll look at that, because looking at the entire connectome across the Drosophila brain is a little hard to really get any insight out of, at least at this point. So, you want to look at the different connectomes in different ways. Finally, we also examine the connectome describing the V1 visual cortex in mice, and this is available. This is actually much bigger than even the Drosophila connectome, or the subconnectomes. But this is very specific to its function, visual processing, and of course, we just talked about that with Lilith, and how that's has some sort of correspondence, at least these people think it does, to artificial neural networks. And there are a lot of methods you can use to look at that. But what they're doing here is they're looking at all these different biological connectomes and looking for this computational motif. We conclude by discussing the potential relevance of this very simple XOR motif as the core piece for configuring biologically viable computational neural networks. So this previous commentary is referenced to, and that's here, implementing Engrams from a machine learning perspective. XOR is a basic motif, and this is also an arXiv paper. This is the paper here. So this is where they kind of get into this using the single model in C. elegans, and then kind of thinking about autoencoders as sort of the comparator for this. So that's maybe worth reading as well. Okay, so this is an image of the basic XOR motif. This is where we have our different sort of cell types and connections. So we have, I guess, our E1, E2, E3, and E4 cells, our excitatory cells. And then our inhibitory interneuron, which is in the middle, INH. And then the output at the right, which is XOR. So we have two different types of connection. We have the excitatory connection in green, the inhibitory connection in red. E1 goes to, and it sends an excitatory connection to the inhibitory interneuron and E2. E3 sends an excitatory connection to the inhibitory interneuron in E4. Then the inhibitory interneuron, as it's activated by the excitatory connection, sends an inhibitory connection, shown in red, to both E2 and E4. Then E2 and E4 sum those excitatory and inhibitory connections, and then send this excitatory connection, both of them, to XOR. So it has to be the same state going to XOR to be activated. So if this, and then like we talked about with time delays, the time delay might play a role in this, because if the inhibitory signal is sent before the excitatory signal to E2 and E4, it might result in a different outcome to the XOR node and, produce a different outcome. It may not, say, for example, produce a non-zero output to XOR. So that's the structure of the motif. What they're arguing is that they find these in connectomes where we have this mix, this balance of excitatory and inhibitory connections, and that they give this output that is then useful to the rest of the connectome. So tiny groups of cells connected in a certain way with certain types of connections with certain outputs. So like I said, they use spiking neural networks. They report their spiking patterns for the XOR motif neurons. Looks like they're using the Brian2 platform for doing these simulations. So Brian is a sort of an electrophysiological simulation platform, and you can use this to model spiking neurons. This is some variations on the XOR motif. So this is a comparison of a basic strict XOR motif on the left and an example of a virtual one on the right. This includes additional connections among the nodes. Strict motifs are defined as an isomorphism between the motif and a subgraph in the connectome, where virtual motifs are defined as monomorphisms. So this is kind of a tricky point about modeling, but isomorphism is where you should have a one-to-one correspondence between, say, like if you're looking at a biological system like a connectome, and you're looking at a model like this, that it should be a strict one-to-one model. You should be able to find this configuration on the left in a biological connectome, and it should give you the same outputs. It's a model, but it should apply to the real world in a one-to-one way. These virtual motifs are a little bit different because you can modify them in the model, and they basically match the behavior of the thing that you're observing in the biology. So here they add a couple, they have these connections. It's a little bit more detailed. They're recurrent connections from, in the strict model, we're going from left to right. In the virtual model, we're going from left to right and right to left with additional connections, additional inhibitory connections, and so on and so forth. So we might see a motif wired in the way that it is on the left, but it doesn't have the same behavior, it has a little bit more complex behavior, and so we might assume that there are different connections or different things going on. And so that's the difference here. So they have these virtual motifs and these stricter motifs that they also identify. Then they propose this different motif here with 6 interconnected neurons, one of them inhibitory, and 8 edges. So this is where we have our same configuration in terms of cells as we did in the strict model, the isomorphic model. But in this case, we have it's wired a bit differently. And, so some of the configurations overlap with the strict XOR motif, but it's a different type of motif. And so you might find different motifs, like if you take a connectome like C. elegans, you might find examples of the strict motif. You might find this as an alternative motif. You might find the monomorphic example as another type of motif. And then, the idea when you look at motifs in networks is that you find these very small groups of neurons connected in a certain way. A lot of times people will do network analysis by looking for things like triangular motifs, or other types of very small sets of connections, that occur again and again in the network. And then they might count up how many of those motifs they find and where they find them in the network. And then that tells them something about information processing in the network. It might tell them something about the topology. It might tell them something about the robustness of the network. And it might tell them something about like the sort of the small worldness or the connectivity of the network. So there are a lot of things you can tell from motifs. It's a very small set of neurons, a very small set of nodes. But if you look at these and they repeat again and again, you can kind of piece together what the global state should look like, or how this small group of nodes affects the global state. Yeah, so then they have an example of fold feedback and asymmetric feedback. Basically, that functionally they can either be sort of symmetric or asymmetric. And then they get it, so that's like kind of from C. elegans, and they're doing this theoretical work. Then they look at the XOR motif in Drosophila, and so now they're extending it to a larger connectome, and they're actually looking, because now you have different types of neurotransmitter that you don't have in C. elegans, and you can they're using these Brian simulations to get at this sort of stuff. They have, now they're looking at different neural pill, and they're looking at the number of neurons in each neural pill, the number of connections, and they actually enumerate the number of XOR motifs, which vary across different neural pills. So for some neural pill, relative to the number of connections, are quite abundant, versus sometimes you don't have any of these XOR motifs in certain neural pill. So that suggests that there's maybe a functional difference between different neural pools. Then they go to the mice. The mouse example, I think, is this one. This is where I guess they. Okay. Okay, so they do some work in mice, and they kind of get into the statistics of that. And so, again, these are very different connectomes, different size connectomes, and we're just looking at the presence or absence of these motifs. And so if we do that, we can actually find them, and they operate, according to the same principles. And so there must be some larger sort of function for these motifs. And so, and again, so they say the analysis of the presence of the XOR motif in very different connectomes from worms to mice indicates a potential interest of this explicit configuration involving excitatory and inhibitory neurons, already suggested by the previous results, considering 3 and 4 node motifs in different connectomes, and also by the importance of this winner-take-all circuit topology. So, however, the analysis presented here proposes that the XOR motif may provide a common basis for better understanding the importance of EI balance, or excitatory inhibitory balance, for homeostasis, and probably its relevance as a direct feedback signal for learning, implementing a kind of basic loss function. So there's a whole literature on homeostasis and EI balance that they are kind of referencing here. And they don't put it in this section, but there are people have looked at this both sort of neurophysiologically and then computationally. Although the relevance of computation at the dendritic level is well established, these XOR motifs may provide the key to computation at the neuronal circuit level. This may be the case in mouse primary visual cortex, where a GLIF network based on the same connectome as we studied provides firing rates comparable to those obtained from biologically detailed models using NEST, which is a platform. The integration of inhibitory and excitatory neurons in the motif suggests a different approach to solving the XOR problem than other proposals using spiking neurons. So this is where, we can look at this with using spiking neurons, but we can look at this using other methods as well. If we're comparing it with neural networks, we can model those as spiking networks, but ultimately, we can use other models to sort of validate this. Okay, so the possibility that a simple regulatory control mechanism can also drive a quite sophisticated task, such as learning, is yet to be confirmed by understanding how much XOR feedback is re-injected and used to promote plasticity. So that's why they were doing these recurrent models as well, just to show that you can have this motif as a static thing, but you need some recurrence to promote plasticity and learning. And so that's why they did that, because they want to show sort of how this would work over time. But it seems computationally feasible based on the well-known results of using random feedback to train computational neural networks. So this is where they kind of go into learning, but then, they stop short of saying that they can demonstrate learning. Nevertheless, I think it's a really nice paper because they kind of get into this cross-species comparison. They get into the motifs and what they should look like, and then they get into the functional aspects as well. So do we have any questions or comments about that?
Sir, I would like to understand what is neuropils. I'm like new in.
So neuropil is any area of the nervous system composed of mostly unmyelinated axons, dendrites, and glial cell processes that forms a synaptically dense region containing a relatively low number of cell bodies. So we have, these groups of unmyelinated axons, dendrites, and glial cells that form a synaptically dense region. So you have cell bodies, their synapses, and it's just this area where um, we can basically get a lot of connectivity. So I mean, this is like, I don't know if like that makes sense, but let's see if I can find an image here. So examples of the neural pill, and this would be in the mammalian brain, is the outer neocortex layer, neural cortex, inner plexiform layer, and outer plexiform layer, and the glomeruli of the cerebellum. These are all found in humans with the exception of barrel cortex, which is found in rats and mice, but many species have counterparts similar to our own regions of neural pill. And of course, Drosophila, it's going to be completely different regions, but basically there are these connection dense regions in the brain. And so they have diverse roles. They sort of serve as way stations for information flowing from the olfactory receptor neurons to the olfactory cortex. Again, we can come back to it later. The second thing I wanted to talk about was this paper. This is shifting gears now to organogenesis and tissue formation and things like that. So this is a paper in Biology Letters, and it's called Metabolic Complementation Between Cells Drives the Evolution of Tissues and Organs. And this is Mihaela Pavlichev, DeFrancisco, and Wagner. They're all sort of evolutionary biologists, developmental biologists. Okay. So let's read the abstract first. Although evolutionary transitions of individuality have been extensively theorized, little attention has been paid to the origin of levels of organization within organisms. That's a mouthful. So basically, they're looking at levels of organization, and they're looking at this, thinking about this kind of how you might think of evolutionary transitions. So, you have individual cells, and somehow those form cell populations. So you have this sort of transition to multicellularity. Then from multicellularity, you go to some sort of functional next step, which is to have organs or have groups, functionally distinct groups of cells. Then you have organs that support sort of a homeostasis. So you have, different cell types in, say, a tissue or an organ. And then you have those organs grouped into organ systems, tissue systems, and then organisms. So you're building up from single cells, and you're building up these layers of complexity. And, that doesn't happen just out of, nothing. There have to be these transitions between, say, like a bunch of cells and then like a population of cells or a multicellular collective. People have studied this in terms of evolutionary transitions, but in development, we haven't really taken that idea and moved it into the developmental realm. So that's what they're going to do here. How and why do specialized cells become organized into a specialized tissue or an organ? What spurs a transition in organizational level in cases where the function is already present in constituent cell types? So, this is where, again, you have this certain level of complexity, and then you're moving to another level of complexity. And sometimes you're sort of recapitulating function. In other words, we are tempted to think that the reason why we have tissues instead of individual cells is because it confers some sort of functional advantage. But it may be that the cells are already doing those things. It's just that forming a tissue might not have an immediate benefit functionally, but it might actually build upon complexity. So it's it's hard to say why these transitions happen, but we first of all want to identify that those transitions are there and then figure out, how they kind of happened and why. So these are the kind of questions they're asking, and people don't really have very good answers for them. So they propose a hypothesis for this kind of evolutionary transition based on two features of cellular metabolism. The first is metabolic constraints on functional performance. And then the second is the capacity for metabolic complementation between parenchymal and supporting cells. So basically, if you have like cells that are sort of doing their job, there are certain metabolic constraints that prevent them from really kind of reaching they reach an energetic threshold and they can't do anymore. They can't really, they have a certain limit to their functional performance. But if you build this sort of multicellular system, this tissue, this organ, then it enhances their ability to do metabolic things, and it removes those metabolic constraints. And then the second, that the capacity for complementation between different parts of the tissue or organ is increased. So this is where we have these sort of cells and then some supporting things that support the cells, and it gives the cells like this sort of ecosystem to live inside, and it increases their function as well, or their metabolic capacity as well. These features suggest a scenario whereby pre-existing specialized cell types are integrated into tissues. When changes to the internal or external environment favor offloading metabolic burdens from a primary specialized cell type onto supporting cells. So this is where we have, pre-existing cell types kind of not integrated into a tissue. Then they get integrated into a tissue. When changes in the environment, you can really have, really highly specialized cell type, and they can exist kind of in a network of cells. And they don't really have to form an integrated tissue. But sometimes you want to offload that from the primary cell types to other types of cells. So sometimes, you want to have like 3 different cell types managing, energy balance, or something like we saw in the last paper. If you need a certain circuit that balances excitatory connections with inhibitory connections, you could have a bunch of cells that do that independently. But integrating that into a connectome, integrating that into brain tissue, might be more advantageous for replicating that motif, or maintaining that motif, and maybe maintaining learning over time. So offloading metabolic burdens is a good way to put that, because doing specialized functions requires a lot of energy, requires a high degree of metabolic stability. And of course, cells are, sometimes they reach a certain amount of metabolic stability. And if their metabolism is too high, they can become cancerous. They can, undergo apoptosis, things like that. So if we can offload that capacity onto supporting cells, we can preserve the cells and keep them from doing bad things. So this really kind of speaks to how, in evolutionary transitions, we often think about, say, like individual cells forming a collective of cells that do something, and they sort of give up certain things about their cell identity to the collective, to different other cells. They offload things. In the transition to eukaryotic cells, for example, there were organelles that would, offload their ability to be a independent organism to the eukaryotic cell, so that they could survive as, specialized units within the eukaryotic cell. So that's kind of what they're thinking here. And that's, of course, an evolutionary transition, which means that you observe this large-scale change in the structure of these cells from, a bunch of cells that are dividing to this functional unit. Okay, so we illustrate this process of suprafunctionalization using the nervous system and the pancreas as examples. So in order to understand this, they talk about different types of evolutionary transition, and that, there's this aspect of looking at this problem as a evolutionary transition. So they actually talk about some examples from biology here. And a lot of times, you have sort of, functions, and you want to acquire new functions. But in a lot of these transitions, you already have that function, and you're just trying to figure out a more efficient way to do it. So they talk about how new traits are associated with a novel function a lot of times in biology. However, many organs perform functions that existed prior to the origin of the organ. So, for example, cells that produce digestive and detoxifying enzymes in the early branching chordates predate the origins of the liver and pancreas. So this is where we have this function of digesting and detoxifying things, and we have those sorts of functions before we have a liver or a pancreas. So liver and a pancreas are basically just more efficient ways of doing this, perhaps, but it involves cells kind of becoming a collective and building this organ. Another example is neural cells that predate the central nervous system, and specific endocrine cells that predate endocrine organs. So before the central nervous system, in basal organisms that didn't have a central nervous system, there are examples that we have today of organisms that have, say, a nerve net, or they express synaptic proteins, but they don't have neurons with synapses, or they have cells that communicate electronically, but they're not neurons. Those all sort of perform the functions of, like, neurons or network neurons that are in a central nervous system, but do very similar things. So a central nervous system is an innovation that occurs at a certain point in evolution. You have sort of, remnants of the ancestral condition where you don't have a central nervous system, but you have other solutions to that, so the question is, why did we evolve a central nervous system, and why do we see it in so many organisms? And the answer is because of this sort of evolutionary transition. There was some evolutionary transition at some point in evolution, and it was successful in terms of processing information and providing this sort of means for behavior. So that's, again, another example. And then, of course, the endocrine example. So these observations imply that the origins of some novelties are not associated with the origin of the function they perform. So that means that the origins of the novelties, the origins of these systems, of these specialized tissue systems and organ systems, aren't really associated with function. Instead, they just arise by the integration of functionality. So, in other words, it's not the function itself, it's integrating the different cells into functionally integrated networks. So, again, there's this higher level unit that forms. Maybe it does things more efficiently, but there is this offloading between cell types, so that's over time, certain cell types become specialized for what they do best, and then they operate in this integrated unit. And again, I think eukaryogenesis is probably a good example of that at the cellular level. So you have organelles that form a cell that's, sort of this evolutionary transition from, say, prokaryotic cells. So this raises new questions. How and why do specialized cells become organized into a tissue or organ? What spurs a transition from cells to tissues when the function is already present? So these are two questions that you might ask. The first is that how do you get these specialized cells to become associated and into this integrated tissue or organ? And the answer to that might be that they're living in association and they eventually form this unit, and it just becomes sort of this integrated, tightly integrated thing over time. And we don't know the answer. That's what we're trying to find out. And then what spurs a transition from cells to tissues when the function is already present, which is a good question because you don't really have, maybe there's selective pressure for maybe a more efficient system, but not for that system itself. So it's really kind of we want to figure this out. We kind of have to model a system of cells and present these different scenarios. They talk a little about metabolic constraints, how there are these metabolic constraints on single cells, and having this population or group of cells acting collectively can reduce these constraints for any one cell type. So cellular metabolism, which are the basis of these metabolic constraints, refer to chemical reactions providing free energy and biomass. Each cell can only generate a finite amount of cellular energy by utilizing a limited number of metabolic pathways. Therefore, augmenting a cellular function or introducing a new function reduces the metabolic resources available for other pathways and cellular needs such as self-maintenance. So this is a stoichiometric constraint. Stoichiometry is just the balance of different molecules and balance of their production. So an example of a stoichiometric constraint is where you have a molecule of glucose which can only be used once. So when you have when you have a store of glucose molecules, say you have 1,000 glucose molecules, you can only use those once, so you only have so much glucose for 1,000 reactions or so. So this is the idea that you can't do any more work given the amount of metabolism you can do. And so if you're living as a single cell, then there's that constraint. If you're living in an organ or a tissue, then you can distribute what the requirements across different cells. So the stoichiometry essentially becomes, or the count of molecules becomes larger, and you can do more. Work apparently. So such trade-offs are often invoked to explain the metabolic shifts in energy and biomass production and high glucose and oxygen availability in unicellular organisms and tumor cells. So eukaryotic cells invest most of their metabolic resources in self-maintenance and performance of service functions rather than biomass increase. So really whereas in a single cell, you might do more reactions to become larger. You might, in eukaryotic cells, you would use your metabolic resources to maintain yourself or perform service functions. So this is the difference between, say, some other cell type and a eukaryotic cell type. So this is the difference there with that evolutionary transition. So we can maybe assume that that tissues and organs do a similar thing. So they're going to give some examples here where you have a parenchymal cell, which is here on its own. There's this metabolic self-maintenance on the x-axis, function performance on the y-axis. This isn't like a study, but they're just showing the relationship. There's this line that shows death, so you have to do a certain amount of metabolic self-maintenance to stay alive. So that's everything to the right of this dotted line. And then functional performance goes along this red curve. So there's an optimal point, which is to keep the cell just alive enough to be alive, or you have to do sort of just enough metabolism to stay alive. And that's your optimal point. You don't want to do too much metabolism because you don't want to go over the edge of stability, but you also want to stay alive. So this optimal zone is right here in blue. So that's for a single cell. And then for, say, if a parenchymal cell, comes into some sort of association with a supporting cell. And then you see this in the brain a lot. You see this in other tissues and organs, where the parenchymal cell is using the supporting cell as a means for increasing its metabolic capacity. So the parenchymal cell is no longer having to do all the metabolism by itself. It's exchanging energy with the supporting cell, exchanging biochemical reactions with the supporting cell. And you have that same relationship with death as the dotted line, and then this new line, which is effective metabolic complementation. So that means that it shifts this blue function rightward so that the entire function is outside of the zone of death and the functional performance increases. So the optimal level of metabolic self-maintenance is just above keeping those two cells alive. And you end up with a higher functional performance as a result because this curve is shifted rightward. This is something they call suprafunctionalization. So it's where you have a single cell that has a certain sort of metabolic curve. And when the cell is in association, the metabolic curve shifts so that it has a higher operating capacity. Okay. And so then from there, they build a hypothesis for the origin of tissues and organs. And they give some more examples here. And they get into something called trade-offs, which is where, especially metabolic trade-offs, which is where you have different types of, you invest in one thing versus another, like you invest in one function versus another, and you try to balance those out. So, there's a whole literature on the evolution of trade-offs, which is where you have competing fitness imperatives and you try to balance them out. So this is where they say a compelling case of a metabolic trade-off between adaptive function and cell maintenance pertains to the generation of ROS. Metabolically high-performing cells require a large amount of energy. Since the oxidative processes of ATP production generate a high ROS load, this increases the cost of maintenance. Augmentation of focal cellular function is likely to decrease the ability to invest in a cell's maintenance, while the need for repair increases due to the damaging effects of ROS. These are free radicals in the cell. So this is where you're producing a lot of energy, you're producing ATP. There are these oxidative processes that occur, and you get more free radicals in the cell, and the free radicals, of course, can be damaging to the cell if you have too many of them. So you want to maximize your ability to produce energy, to produce ATP, but minimize the effects of ROS. And so, how do you do that? So that's where we're getting this trade-off. And so then that's an explanation for having organs and tissues, is you can perform that trade-off without too much of a cost. So this shows the exocrine pancreas and its evolution, illustrating the proposed transition from individual cells to tissues and organs. So in example A, which is up here, this is an early chordate, and this is Amphioxus acinar cells, which are here, are individually dispersed throughout the intestine. Eventually, acinar cells, which are in the intestine of the Amphioxus here, you can see these dots. It's kind of hard to see, but each one of these little dots down here is a cell. Eventually, acinar cells were integrated into a tissue or organ structure where they are dependent on metabolic complementation from supporting stellate cells. So down here, we have the acinar cell, which is, we see that here as a single cell type in Amphioxus. But in, say, like rodents, you have the acinar cell, which is supported by a stellate cell, which is below. So that provides this support mechanism. And so they're no longer dispersed throughout the intestine. They're integrated into this organ structure where they're dependent on the metabolic complementation of these stellate cells. So you can see that there's a difference that you have in one case with a single cell type. It has a certain metabolic capacity. When you get into vertebrates and especially mammals, you get this organ structure where you have this stellate acinar cell symbiosis, or I don't know if it's a symbiosis. It might be some sort of dependency where it increases its metabolic capacity. Okay, so that's pretty good. And so they give some more examples of tissue-sustaining metabolic interactions. These are just examples from across the tree of life, I guess. They give one example from the nervous system of organisms with a central nervous system. Glial cells play supportive roles, providing nutritional and homeostatic support for neurons. Astrocytes of the vertebrate central nervous system are a paradigmatic example of a support cell. And so they talk about how if you have a nervous system, actually if you have neurons are very energetically costly. And neural activity is also costly. So, it's often said that the brain is like kind of consuming most of the glucose in the body because, it's expensive to have action potentials. It's expensive to be a neuron. And so, you have to think about that when you have bigger brains. When you start to expand the nervous system, you're expanding not only the ability of the organism to have, a brain to think with, to solve problems with, to see the environment with, or to hear the environment or touch the environment with, but you're also increasing the metabolic costs of that to the organism. So this is where you need to have a nervous system. When you get enough neurons, you need to have a nervous system with astrocytes and with other supporting cells. So in this case, astrocytes supply lactate for neurons to use in oxidation to yield APDP, which is involved in energy production. Dependence of neurons of aerobic metabolism is manifest in cultured neurons that die very quickly without oxygen. So neurons are dependent on aerobic metabolism, and you can observe this in culture. Astrocytes store glycogen to overcome periods of low glucose. So astrocytes are specialized to sort of provide a means for aerobic metabolism for the neurons. So that's why they're often called support cells. Uptake glutamate from synaptic space to limit its transmitter activity and synthesize glutamate via alpha cataglutarate in the citric acid cycle to extrude in the intracellular space as a source of neuronal glutamate. So when you couple astrocytes with neurons, and especially neural activity, astrocytes release precursors to glutathione, a major antioxidant effector, strengthening their protection against ROS. Together with endothelial cells, providing oxygen and glucose, neurons and glia form a metabolic unit. So neurons, glia, and endothelial cells all form this metabolic unit instead of just having neurons trying to produce match all of their own energy needs. So that's a good example, and they go a little bit into more detail about that, as well as parenchymal cells in the pancreas. And that's pretty much the rest. The end of the paper. They have some nice references in here, and I think it's a decent hypothesis. It's something we don't talk about when we talk about tissues as much, but energy production and about metabolism, and especially those evolutionary trade-offs that need to be there. Because it's worth mentioning that, when we talk about complex systems in biology, we're talking about different fitness imperatives. We're talking about balancing those fitness imperatives, especially as we have this functional unit, which is the tissue or organ. And, how do we get from, say, like very simple morphologies, very simple systems to more complex systems? So any comments or questions?
I think that's interesting. I want to look into it a bit more closely. I'm still putting lunches together here, but yeah, maybe also think just a little bit about the difficulty identifying cell types in some structures, and you know, like you said, you know, it's yeah, that makes sense if you understand, you know, how cells do change. That there, yeah, like how much pressure there is really, or certainly that you'd expect a spectrum, and uh, uh, just wanted to say, I mean, thinking a little bit about the kind of um, energetics and other things. I went down a little rabbit hole of biological condensates last night.
Oh, yeah.
Just talking about, yeah, and you know, that certainly just acknowledging that we still, there's still a lot of kind of the forces and complexities within a cell that are still very not well understood. Um, and you know, the fact that we're still, say, discovering um, these kinds of, you know, phenomena that at first are discoveries and then are second are found to be absolutely, you know, ubiquitous.
Yeah.
It's one, it's a discovery, like, oh, this is fairly novel, and then it's like, oh, it's actually everywhere we look for it now, you know. And that, yeah, just that you know, some of these pressures and things might be better understood if we understood how even a single cell manages the complexity that it manages. And again, like condensates just being one of those examples. Um, yeah. Anyway, I had a little thought about just, you know, excitatory inhibitory balances from the last paper, but I can't remember that far back.
Yeah.
So I'm catching up on last week's video, but as well. But anyway, great papers as always.
Well, thank you. Yeah. Yeah. So yeah, it's worth thinking about these papers together in terms, especially in terms of EI balance and some of those networks, and then thinking about building complexity and how that happens. And we haven't talked about a lot about evolutionary trade-offs, but it is a fascinating concept because we tend to think of like sometimes we tend to think about evolution as like we think about single fitness imperatives. Like we say, okay, well, obviously there was this trait that was favored, and if we think about this in terms of like the entire organism or in terms of fitness landscapes, oftentimes we have competing imperatives pulling at the organism. So at the organismal level, we have multiple fitness imperatives at the same time. You're in one environment, and there might be one function that is highly favored, but there are other functions that are maybe also favored or disfavored, and so those all kind of tug the organismal population in different directions. And you also have different types of processes, like where you need to produce a lot of energy, but you also need to get rid of waste, or you don't want to you want to produce a lot of energy because you can't have a bigger brain if you don't produce more energy. So how do you solve those problems? And it's not, I don't want to say that cells are actively solving problems, but there are like ways that things that get favored over other things, and that's kind of how we get to this next level of organization where we get specialized tissues from things that are less specialized. So that's kind of the lesson there. But anyways, I welcome people to think about these topics, and if you're interested in coming up with a hypothesis or project of some type or some other thing, you're welcome to present in the meetings, and or even if it's just kind of unorganized thoughts, that's also welcome. We usually chew on those for a while as well. So thank you for attending today's meeting. Anything else before we go? Okay, if not, thank you for attending. See you next week.
Thank you. Take care.
Thank you. Bye.
Nice.
Yep.
Bye.
Bye.
Bye.